Good morning, dear students. Hope that you all are safe at home. Okay, today we are going to study about embryonic stem culture. You all know what is this embryo and what is culture and also stem cell. But anyway, here we are going to study in detail about the stem cell culture. So, by the way, what are stem cells? The undifferentiated animal cells that are capable of proliferation and differentiation to do specialized functions are called stem cells. Okay, so it's a primitive cell with the ability to self-replicate. It can reduce inflammation, it can fight against cell death, it can differentiate into multiple tissues like bone, muscle, cartilage, fat, etc. And then basically, there are two types of stem cells. What are they? The first one, embryonic stem cells. The stem cells come from human embryos that are 3 to 5 days old. And the second type is adult stem cells. It's also called non-embryonic because these cells are taken from the adult. And what do these stem cells do? These stem cells provide new cells for the body as it grows and replace specialized cells that are damaged or lost. See, sometimes our body gets some damages or injury, or some cell may get lost due to some problems. So, in that case, these stem cells provide new cells for the body in that particular damaged place, damaged tissue, or lost tissue. So that is what these stem cells do. And these stem cells has two unique properties. First one, they can divide over and over again to produce new cells. And the second one, as they divide, they can change into the other types of cell that make up the body. And what is a stem cell culture? Culturing of stem cells. Simply we can say culturing of stem cells. And these stem cells are the roots from which different cell lineages arise, and they exist in multiple forms. And these stem cells are cultured using specific transcription factors or chemicals. Then what are embryonic stem cells? So in this PowerPoint presentation, we are going to study in detail about embryonic stem culture. So you should first know in detail about embryonic stem cells. As I have already told you, they are undifferentiated cells isolated from the embryos at blastocyst stage. They are called embryonic stem cells. And these cells are capable of continuous proliferation. They can multiply continuously and differentiation, leading to the development of many different cell types as in the adult. And here are a few characteristics, some important characteristics of embryonic stem cells. The first one, they are found as the inner cell mass in the blastocyst. Then the second property or characteristic, they can divide continuously without differentiation in the culture. And the third one, they maintain usual diploid number of chromosomes. Then the next one, they form clones, mass of identical cells. Then the next one, when embryonic stem cell is injected into any tissue, it integrates with that tissue and becomes the part of that tissue. These are few characteristics of stem cells. See, these embryonic stem cells give rise to specialized cells according to the germ layer from which they are taken. See, from the embryonic stem cells also, there are different germ layers. See, endoderm cells, ectoderm cells, and mesoderm cells. See, endoderm mass of cells. See, if we take these cells from the endoderm mass, from that we can get the cells of thymus, The cells of thyroid, parathyroid, urinary bladder, respiratory tract, etc. will be formed. And then, if it is taken from mesoderm, it gives rise to cells of bone marrow, blood cells, adrenal cortex, muscles, heart, so just like that. And then, if we take from ectoderm, that will give rise to cells of skin, nerves, connective tissues of head, eyes, ears, and so on. So, this is how these embryonic stem cells are specialized. They are specialized according to the germ layer from which they are taken. See, from this picture you can understand better. These are the inner mass of cells. Okay. Endoderm mass of cells, then mesoderm and ectoderm. See here, these are the ectoderm cells, and here is the mesoderm cells, and these are the endoderm cells. So all these together called inner mass of cells, embryonic stem cells. Okay, and then you should also know what are adult stem cells. Here we are not going to study detail about adult stem cells now, but anyway you should know what these adult stem cells are. The stem cells isolated from fully developed organs are called adult stem cells. The stem cells isolated from fully developed organs are called adult stem cells. They can be isolated from bone marrow, bloodstream, cornea, retina, liver, skin, muscles. Okay, can be isolated from any of these organs, but it should be from the adult. If a part of these organs are injured, the adult stem cells regenerate to heal up the injury. So in case if we get any injury in these parts, with the help of the adult stem cells, we can regenerate so that the injury can be healed up. Okay, so they regenerate the tissues lost due to injury, tear, or maybe any diseases. So this picture will give you a very clear idea. See, the adult stems can heal up the injuries in various organs. Okay, then now we'll pass on to the detailed study of the embryonic stem cell culture. As you know, the rearing of cells of inner mass of blastocyst in vitro is called embryonic stem cell culture. These are the various steps involved in embryonic stem cell culture. Step 1, step 2, 3, 4, 5, so on. So various steps. So now we'll see them one by one. The first step. Stem cells are isolated from embryos at blastocyst stage and grown in a suitable chemically defined medium to produce stem cell lines. Okay, so the stem cells are isolated from embryos at the blastocyst stage. They are then grown in suitable chemicals to produce the stem cell lines. So for this, what type of cells we have to take? I mean, what should be the age of the embryo? The embryonic stem cells are obtained from 3 to 5 days old. Okay? 3 to 5 days old embryo can be used. Then step 3, to get human blastocyst, the egg is fertilized with the sperm by means of in vitro fertilization. So, the egg should be fertilized. So, step 3, we need an fertilized egg. So, through in vitro fertilization, it can be done. And then the grown up blastocyst is then kept under the microscopic field, and the inner mass of cells is taken out through a micro needle. So the inner mass of cells is taken out through a micro needle. Okay. And then step 4, the cells are poured into a plastic tube containing a nutrient broth. Okay. So now the cells taken with the help of the micro needle from the blastocyst, I mean from the inner mass of cells, are now poured into the plastic tube containing nutrient broth. Okay, so yes, the plastic tube containing nutrient broth, and the inner mass of cells taken is now introduced into the plastic tube. That is step 4. And then step 5. The inner surface of the culture disc is coated with UV irradiated mouse embryonic skin cells. Okay, so after step 4, what we have to do? You have to take a culture dish, and the culture dish should be coated with mouse skin cells, and this is called feeder layer. The mouse skin cells is called feeder layer, and the main purpose of this feeder layer is that it will not divide, but they provide a sticky surface for the attachment of the embryonic stem cells. They provide a sticky surface. See, this feeder layer will not divide at all, but its purpose is it provides a sticky surface for the attachment of the embryonic stem cells, because it is in this dish we are going to pour the embryonic stems present in this tube. So that we will see in the next step. Okay. So step 6. The cells with the nutrient broth which is present in the tube are poured into the culture dish, and the culture dish is now incubated at 37 degree for several days. So this is the petri dish coated with feeder layer. Now we have to pour the embryonic stem cells in that tube, and this should be incubated for several days under 37 degree centigrade. So now what will happen? These cells inside the petri dish form a mass of cells which will proliferate and form crowds of cells in the culture dish. So when you keep it under incubation at 37 degrees C for several days, the inner mass of cell will proliferate, and now it forms a shape like this, a crowd of cells in the culture dish. Okay, and now step 7. What you can do? You can remove this crowd of cells gently and cut into small pieces with a sterile knife. These mass of cells should be removed from the dish and cut into small pieces with a sterile knife. And step 8. These cut pieces are then inoculated into fresh culture dishes containing nutrient broth for subculturing. See, already we got a crowd of cultured cells. And once these cultured cells are carefully cut into small pieces, and again these cells can be subcultured. Again you take more dishes, and you can culture these inner mass of cells again. So you will get millions of embryonic stem cells. Okay, so that is the step 8th step. Okay, and finally the embryonic stem cell can proliferate continuously for over 6 months without differentiation. So if we just leave these inner mass of cells to proliferate, it can proliferate continuously for over 6 months without differentiation. So this is how the embryonic stem cells are cultured, and this forms the cell line. So that is said to be embryonic stem cell line.